If Graham Hancock's lost civilization actually exists, what proof should we expect to find? Let's ask the two Milos.
The most important piece of evidence that you could possibly have, the be-all, end-all to prove that your lost civilization exists, is DNA.
The best source of evidence for connection between different peoples is going to be DNA. I think we would see a lot of genetic evidence for that, not just in ancient human remains, but still within us today.
Even in the living record, huh? Well, that's funny, because when it comes to old world DNA found in Native American people, which is exactly the kind of thing you would expect to find if Hancock's lost civilization was a thing, it's assumed to have come from post-1492 contact and is removed from the findings. They won't find the genetic evidence because they throw it out.
Okay, let's examine these claims. Delve into the study of modern and ancient DNA and see what this tells us about the hypothetical, lost advanced civilization of the Ice Age. Welcome to Ancient Puzzles. Imagine there was a lost advanced civilization during the Ice Age that was affected by a huge cataclysm, and the survivors tried to restart settling among hunter-gatherer communities, relying on their survival skills and giving them knowledge. This is proposed by Graham Hancock and certainly convinced a lot of people that this makes sense for the most part. He claims members of this lost civilization reached many or most parts of the world, but this is not accepted by the scientific community.
I think that what happened there during the Younger Dryas is extremely mysterious, and I don't think we have the whole story. And I'm simply proposing that the survivors of a civilization who were in very small numbers, traveled around the world seeking refuge, sharing their knowledge with those they took refuge amongst, and sharing the knowledge of those they took refuge amongst. It was an exchange, not a one-way trip, and they did not bring plants and seeds with them. They worked with what was locally available, and that's precisely what we see happening after 12,900 years ago.
Hancock says the survivors were small groups of people, but he also discussed maps based on older sources that he believes indicate someone was mapping the world during the Ice Age, and this must have happened before the cataclysm, when the lost civilization was at its peak.
The problem is that Antarctica appears repeatedly on these much older maps, and it appears in the right place and a bit bigger than it is today, but very much as it looked during the last ice age. So what all of this suggests to me is that the world was mapped and explored by a global seafaring culture with a level of technology that was at least equivalent to ours at the end of the 18th century during the ice age.
It seems possible that only small groups survived, but it's hard to believe that the ancient seafarers who mapped the world before did not settle anywhere in large numbers.
Graham Hancock is the most popular alternative history purveyor in the world right now. And what does he say? In his book, America Before, his most recent book at the time of the making of this video, he writes, "My broad conclusion is that an advanced, global, seafaring civilization existed during the Ice Age, that it mapped the Earth as it looked then with stunning accuracy, and that it had solved the problem of longitude, which our own civilization failed to do until the invention of Harrison's marine chronometer in the late 18th century. As masters of celestial navigation, as explorers, as geographers, and as cartographers, therefore, this lost civilization of 12,800 years ago was not outstripped by Western science until less than 300 years ago at the peak of the Age of Discovery." This is why, during his debate with Flint Dibble, Hancock did not deny it when Flint says Graham believes the civilization was globe-spanning. And this explains why Graham Hancock wants archaeologists to look for evidence of this lost civilization in the Amazon and the Sahara and the coasts around the world.
Hancock claimed his lost civilization was already global during the Ice Age, but Dan did not agree when I discussed this with him time ago, and this is what he told me. Dan was asking me to provide a quote while not providing one himself, but the quote proving him wrong certainly exists, and we know Hancock believes there were sites that date to, or were planned before the cataclysm, which implies the lost civilization had already expanded.
But let's look at sites that are considered to actually be from the time of this civilization. Where are they? Gunung Padang in Indonesia. This is believed to have a pyramid from more than 20,000 years ago, from the time before the cataclysm. In Magicians of the Gods, Graham Hancock refers to Sumerian documents that speak of cities being built before the flood, and suggests that he thinks these cities in Mesopotamia existed then. I know Hancock does not believe that the Great Pyramid of Giza was built during this period, but he does believe it was planned out in this period. Dwarka in India is thought by Graham Hancock and many others to be not a city that was built by survivors of the cataclysm, but a city that existed before the cataclysm.
Seems like Dan misrepresented Graham's position to score points. But even if we ignore it and assume Hancock never said they settled anywhere during this period, that doesn't mean it's okay, because it's certainly very strange and unrealistic. To me, assuming Hancock is right about everything, what makes more sense is that part of the people pseudo-archaeologists often call primitives, or more likely their descendants after admixing with the newcomers, became members of that civilization before the cataclysm, while others still chose to live as hunter-gatherers that Hancock does not deny also existed. But no, Dan wants us to believe the people of this lost civilization didn't settle in the lands they discovered and mapped, and that before the cataclysm, there were little or no interactions with the local populations. Unfortunately for Dan, Hancock has not retracted any of that yet, although it's true that during the debate with Flint Dibble, he focused more on the small groups that supposedly migrated after the cataclysm, but a lot of details on what else was this lost civilization doing while drawing world maps are missing. They possessed advanced knowledge and great seafaring skills, and seems fair to assume they had agriculture and megalithic architecture, long before they started to explore the world.
But then what was your civilization growing?
I don't know.
Hancock believes that his lost civilization practiced agriculture, but when it comes to metallurgy, he seems less convinced, a stance that appears inconsistent with what he wrote in his books.
I'm suggesting that all these memories tied up in ancient myths are memories of real events. These were the survivors of this great lost civilization. I'm suggesting that all these memories tied up in ancient myths are memories of real events. These were the survivors of this great lost civilization.
If Hancock pretends that what some sources say is good evidence to support Ice Age agriculture, which he tells us was transferred by the survivors of the cataclysm, the same should apply to metallurgy. It's clear in his work that agriculture and metallurgy are mentioned in the same context. So either the material he selected is not good enough to back up his claims on agriculture, or his lost civilization must have had agriculture and metallurgy.
The issue of metallurgy more than 12,000 years ago has never been one that I've pursued in my quest for a lost civilization of the Ice Age. Whether or not metallurgy was being done somewhere on Earth during the Ice Age isn't a fight I have a dog in.
Hancock backpedals on metallurgy, seemingly unaware of the implications for his claims about agriculture, a development or idea, as Graham said during the debate, that it's hard to explain wasn't brought to distant places until the Younger Dryas period. Besides misrepresenting Hancock on social media and YouTube, Mr. Dan the DeDunker also shared his own version of the lost civilization, which, based on his content, is not very different from Hancock's proposal. He disagrees on when such an advanced society spanned the continents, though he doesn't admit it, and doesn't believe in lost ancient high technology or psychic powers, but buys there was a cataclysm, small groups of people spreading knowledge, and takes the idea of world maps during the Ice Age very seriously. Many features can reveal how advanced a culture or civilization was, but the global aspect seems to be something everyone agrees qualifies as advanced, especially if the people must have had advanced seafaring skills in order to reach distant lands. The concept of civilization, by the way, is commonly used by people, but archaeologists, historians, and anthropologists tend to not use it because the definition changed many times, and it's not considered useful to describe past societies. DNA alone is not helpful to determine if ancient populations used power tools or hand tools, but it can definitely tell us where they came from and their distribution. If a specific group of people was widely distributed, not necessarily occupying every single region, it should be possible to find the DNA evidence someday. But Dan concluded that this will not happen and blamed the methodology he thinks geneticists use. He retracted most of what he said in the video where he discussed this, but his behavior was still terrible, and I highly recommend you to watch this for more details. He at least did something right by sending me the video, and thanks to that, we all can finally learn something from it. This is what I'm going to explain, which yes, I know it's a lot, but I hope you'll find it useful and interesting, and of course, I'll mainly use that video to do so, but I will also refer to other content he posted on YouTube and social media as well. Based on the thumbnail, one gets the impression that his argument is more about Atlantis, but the right word is actually Atlanteans. DNA can't tell us if the lost city of Atlantis existed, but if we accept the possibility of a lost group of people reaching many parts of the world in the very distant past, it is reasonable to think there's a chance to find human remains and get the DNA. Proving something is definitely possible with good evidence and results replicated multiple times, but that does not always imply other theories or hypotheses can't be true. Using DNA research pretending it puts something to bed can certainly be problematic in some cases, even though the more evidence we have supporting a specific conclusion, the less likely other options will be. Mr. Dan the DeDunker admitted there were lots of problems in his video, but he moved the goalposts and still pretends something weird happens in the Americas, so we're going to see that geneticists can achieve, and have achieved, a lot more than what he says. These claims basically affect two fields: human population genetics and archaeogenetics. The former is a subfield of population genetics, which itself is a subfield of genetics, and the latter is a subfield that studies ancient DNA, but it's not restricted to humans, since animals and plants are also included. Those are also important to find evidence of a lost civilization, but in this video I'll be focusing on human DNA. In both cases, the subject of study are specific locations of a person's genome, alive or deceased, and even though the difference between genetics and genomics is well described, the term population genomics is often associated with population genetics, and it's definitely used in some ancient DNA studies, which is what I have mostly read. The difficulty to study a single gene without also studying other genes, as well as other factors that influence the expression of a specific set of genes, like the environment for example, make these two branches of molecular biology virtually synonymous, and thus explains why the terms are used interchangeably. That said, let's now learn the three principal types of genealogical DNA tests that matter to find evidence of a lost civilization: Y-DNA, mitochondrial DNA, and autosomal DNA. Y-DNA is passed down from father to son, so only males have it. Mitochondrial DNA is passed down from mother to child, and both males and females have it. These two cannot be used for ethnicity estimates, but can be used to find one's haplogroup, which can be further divided into subclades. We often know where the different haplogroups originated, or most likely originated, in the ancient populations who carried them, but to estimate admixture proportions, autosomal DNA is what we need. The autosomes are inherited from both parents, and we will see Dan didn't take this into account, either cause he did not understand what he was looking at, or conveniently ignored it, since he cited papers including admixture analysis, but drew conclusions that don't make any sense. Also, PCA is commonly used in genetics to infer and visualize population structure and admixture between populations, and we see it in some of the papers cited by Dan as well. There are lots of concepts in genetics, but I don't want this to be too long and maybe boring, so I'm going to finish this part highlighting the following, since it's more directly related to the papers I'll be discussing. Ancestry informative markers are primarily the single nucleotide polymorphisms that exhibit substantially different frequencies between populations, and these are mainly used not only for ancestry or in evolutionary biology, but also for the detection of disease risk factors. Short tandem repeat analysis is a method used to compare allele repeats at a specific locus in DNA between two or more samples. Y chromosome, X chromosome, and autosomal short tandem repeats are commonly used in personal forensic identification and kinship testing, even though SNPs can also be a good complement or useful on their own when analysis of short tandem repeats fails. STR markers are also commonly used to estimate Y haplogroups, but they're less reliable than SNPs for that purpose, although they're still a powerful tool that shouldn't be ignored since it's useful data to compare, and sometimes can even provide lineages within haplogroups. We can describe SNPs as common genetic variations found in a sufficiently large fraction of the population, generally 1% or more, while short tandem repeats are highly variable and mutate much more frequently. The idea is that both markers are important, but it's not rare to find papers focusing solely on STRs. Another way to determine if there is a close genetic relationship is the detection of long runs of homozygosity segments. Basically contiguous regions lacking variation in the genome, so when long runs of homozygosity arise, the only plausible explanation is that the person inherited two copies of the genome from parents who are closely genetically related. Identity by descent segments also matter, which should not be confused with identity by state segments, as the latter certainly implies having the same nucleotide sequence, but not necessarily inherited from a common ancestor, unlike IBD. Several existing methods identify these segments for single nucleotide polymorphism array, or whole genome sequence data, including a few that can detect them in ancient DNA data. Speaking of data, this is what it looks like, with each letter representing one of the 4 nucleobases found in DNA. Adenine pairs with thymine, and guanine pairs with cytosine. RNA is slightly different because, instead of thymine, it contains uracil. Finally, the most recent common ancestor of a set of organisms is the most recent individual from which all the organisms of the set are descended, and this is also used in reference to the ancestry of groups of genes, haplotypes, rather than organisms. The time to most recent common ancestor can be estimated based on DNA test results and established mutation rates, as practiced in genetic genealogy, or by reference to a non-genetic, mathematical model or computer simulation. Dan's main video can be divided in 2 parts. In the first one, Dan makes clear it is possible to disagree with that, but he's actually preparing the viewer to accept his final piece of evidence as fact. He brings papers that are not very relevant to back up his main argument, but he uses them to show that this kind of research has limitations. That isn't necessarily a bad thing, but poor research, cherry-picking, and misrepresentation can definitely make a difference. The second part is all about one paper, used by him as the smoking gun evidence supporting his point that it doesn't matter if geneticists have the evidence in front of them, because the way things are currently done excludes the possibility of an Ice Age lost advanced civilization as a rule. We'll get to that, but it's important to check the first part as well, because there we'll see Dan's knowledge on the subject matter was very limited.
And that's just like across the board. That video was outdated. And that's the first thing I want to mention is this is information that was outdated because I already knew it. So when I went and looked the stuff up, I just, like, research bias, I guess you would call it. I knew what I was looking for. I knew these facts. I already knew this stuff. I've known it for years. Well, that's why it's outdated, Dan. But I was reading that kind of stuff 15 years ago. That was cutting edge knowledge to me when I was in my mid-30s. So again, I'm sorry. I was wrong, and this is not an excuse to say that. I'm just pointing it out to say I did not deliberately misinform anyone.
Dan forgot what he posted on social media, which confirms he couldn't know many of these facts 15 years ago as he claimed in the DNA retraction, So there were obviously much bigger problems than the outdated information. His dishonesty and lack of understanding.
The first thing to mention is ancient DNA can't be used to establish a dating framework. This seems to be something people believe without really looking into it, but it's clear when the DNA is tested, dating comes out from outside the field. They'll carbon date samples to get an age estimate because the genetic material offers us little in that regards. Studies closer to our time can do a lot better with dating when they have several samples from a family and can use those to establish a firm tree. They can extrapolate and figure out the dating pretty well in a few generations around that. But that is not the case with ancient DNA. When they find hundreds of samples from the same time and place, they still have to date these samples from outside the field of genetics. It's just simply not equipped to do the job. There is one way it can help with ancient timelines. It can tell us which group came first when it comes to haplotypes. Which group begat the other group is something that it can discern.
History and archaeology can help geneticists to interpret the data, and if archaeologists don't find human remains, there's no ancient DNA. Simple as that. Archaeogenetics is part of both genetics and archaeology, even though it's fair to say that many of the scientists in this field are not trained archaeologists. Geneticists obviously play an important role, but so do mathematicians or computational scientists, for example, meaning that the approach is interdisciplinary, and I'll refer to all of them as geneticists or scientists for the sake of simplicity. That said, archaeogenetics relies on the meticulous work of archaeologists, who document all the details to provide context and determine the age of the remains, tasks that are essential. When it comes to haplogroups, we can of course know which mutations came first, and it's typical to calculate the time to most recent common ancestor, which notably is not the same as the actual date of emergence, but we will see how gathering lots of ancient DNA samples can greatly help in figuring out where and when the different branches existed. Dan also missed there are other ways to infer dates from DNA, but not everything was incorrect, since the archaeological and historical evidence obviously matter, but so does the genetic evidence, and previous notions can also be challenged.
Areas that are explored and settled and colonized and even trade routes. Traditionally, these are almost exclusively men that do this, and then they go establish the beachhead, and then they bring in women afterwards, order a wife or something. And I'm not trying to be sexist about this. This is something that geneticists have to deal with because those haplogroups, they're divided by your chromosomes. Now, because these haplogroups are sent down either the male or the female line, a father passing it to his sons and a mother to her daughters, this makes early settling, small settlements, and sporadic trade a lot harder to find in the genetic record. Now, my point is when you think of a small group of people interbreeding with other people around the world, you cut the odds of finding this stuff in half when you say that it's almost exclusively men that showed up there, because every time a daughter is born from that line, the DNA kind of dies off and you can't find that haplotype anymore, which is usually how they track the races and the whole distribution of genetic material through the world.
The wording is confusing, so I'll start clarifying that haplogroups, and the many different branches that arose over time, are of great interest and useful to track population movements, but as already stated, they cannot be used to determine race or ethnicity. Having said that, we start to see a big problem here, which some of you watching probably noticed as well. When Dan made this video, seems like for him haplogroups were the key to solve the mystery, ignoring that, even if the haplogroups of a lost civilization were lost in subsequent generations, we still have something called autosomal DNA, that it's definitely helpful to estimate admixture proportions, and can be compared against the available modern and ancient datasets, being then valid to reveal if a specific group of people reach distant lands. Ancestral components can disappear after many generations since the first admixture event, but we have to assume that the idea of small groups of people is true, and we've already seen the problems in this logic. Also, men carry mitochondrial DNA, so if we're lucky to find human remains of the newcomers and extract their DNA, even if all of them are men, we can know their maternal and paternal haplogroups, including the subclades in most cases, and their descendants will have part of their admixture, no matter the haplogroups they carry. So getting samples of admixed individuals is also useful, and you'll see some examples. Dan cited 2 papers to support what he said and explain to us what geneticists do. In the first one, he highlights this part, but he's done researching the topic. He does not spend time trying to learn how peculiar the sex bias aspect is in Madagascar, or what the scientists see when a genetic contribution is sex-biased, so he was missing quite a lot. What he showed is obviously real, but at the end the study says multiple Indonesian regions may have contributed to the settlement of Madagascar, also acknowledging that sea nomads traveled with their families, even on long-distance journeys. Focusing more on the data, it turns out that the presence of Indonesian mitochondrial DNA in Madagascar was well known when the study was published, ranging from 11 to 50% depending on the ethnic group, which is quite significant in my book. The maternal haplogroup in question is this one, but the Malagasy carry specific point mutation variants, which together have been termed the Malagasy motif, meaning it's simply the Malagasy version of the Polynesian motif, since it's found almost exclusively in Madagascar. And of course, there are paternal haplogroups shared between the Malagasy and Indonesians, and one subclade indicates genetic contributions from southern Borneo to Madagascar. Another study in 2014 had already estimated that the Austronesian gene flow in Madagascar represents at least 30% of the 3 populations they studied, but more research was published after this paper, and in this study, the best fit admixture model obtained indicates the Malagasy carry 37% admixture related to the Banjar, who currently live in southeast Borneo, and 63% related to the South African Bantu population. Similar results were reported in 2017, including an estimate of the West Eurasian admixture that makes sense considering a minor presence of West Eurasian haplogroups, and also, it was found that Y chromosome lineages of African origin are much more frequent in Madagascar than the ones originating in East Asia, in contrast to the mitochondrial DNA lineages. According to the authors, it seems we're looking at strong sex bias involving contributions from Bantu males diffusing from the north to the south, and the evidence suggests that the southeast was already settled by Austronesians, but they were both men and women in this case. Despite the fact it's possible that Madagascar was settled from East Africa by an already admixed Austronesian Bantu population, there is little support for that in DNA studies, that for the most part, agree the Austronesian settlers came first, as part of what it's called the Austronesian expansion, that it is believed to have started in modern-day Taiwan, between 4,000 and 4,500 years ago. Austronesian-speaking groups equipped with new sailing and farming technologies, over a long period of time, also reached the northernmost Philippines, specifically the Batanese Islands, Eastern Island, and dispersed into the islands of the Indo-Pacific, culminating in the settlement of New Zealand in 1250 AD. They almost certainly reached the Americas as well, and when it comes to Madagascar, Austronesians could have settled the island more than 2,000 years ago, but it is likely that other clans came from the 7th century onwards. On the other hand, Bantu peoples may have begun migrating to the island 100 years earlier than these Neo-Austronesians, but the estimates indicate the admixture event took place much later, between 500 and 900 years before present. This can be discerned thanks to many different tools, like those checking the length of admixture tracts to infer dates, or others based on linkage disequilibrium, which given an admixture event, analyze how recombination breaks down associations between alleles over successive generations, leading to LD decay that can be measured and used to estimate the number of generations since the admixture occurred. The methodology was used in this paper published in 2018, which also reported evidence of selection in favor of African ancestry, so the 37 or 38% Asian ancestry would be even higher if this had not occurred, probably around 45 or 50%. Dan chose a paper on a topic that it's a terrible example to illustrate what a male-biased genetic contribution looks like, let alone if the intention is to compare it with the hypothetical non-realistic lost advanced civilization. That, remember, was characterized by small groups, men for the most part, and apparently there was no time for more people to settle before the cataclysm. even though they were supposedly drawing world maps. On one hand, the current evidence suggests lots of Austronesians traveled thousands of kilometers across the sea, settled in Madagascar, and it's not very relevant if they were mostly men at the beginning, because by the time people from Africa came onto the scene, seems like what they found there is men and women in roughly equal proportions. I have to say that there is very interesting evidence of human activity in Madagascar, the oldest dated to 8,500 BC, but we don't know if there were still descendants of any population when the Austronesians arrived. And on the other hand, the evidence supports that the African contribution was sex-biased, but the Bantu-associated migrations are not a good example for two reasons. Their maritime expansion was more limited, and a lot of them definitely came to Madagascar, including women to a lesser extent, as attested by the genetic results. Anyways, what Dan highlighted wasn't even about them, and ancient DNA is what can give us a more accurate picture of the people who inhabited the island in the past, but to my knowledge, we don't have these studies yet, so maybe we'll see something surprising down the line. There is still conclusive evidence that the modern Malagasy people have a significant amount of Austronesian admixture, and there is no reason to think the age of this signal, or who were the earliest inhabitants, will not be properly investigated in more detail, but it will take time. Asian ancestry is more prevalent in the center of the island, while African ancestry increases as we move towards the coast. As for the West Eurasian ancestry, I'll cover it later. Now let's move on to the Neolithic in Europe. Okay, we can see that this 2011 study on the Neolithic cited by Dan mentions the autosomes, but he either missed it or couldn't link the concept with admixture proportions, since in this paper only haplogroups and kinship are discussed. He could have chosen another one on Iberia published in 2022 that he should have known already, because time ago I even shared with Dan a video I made about this research. In the 2011 paper, the authors couldn't determine if the individuals tested were related, unlike this more recent study, where long runs of homozygosity segments were detected. I'm not blaming anyone for that though. Just note that this is very common in the field, which offers an opportunity to investigate aspects that can be difficult to infer using more conventional archaeological methods, and this is not limited to genetic history, as biological kinship and sex also provide details about funerary practices and other cultural aspects, such as determining whether both men and women deserved monumental graves, or proper burials, whether members of the same family were buried together, or whether mating with close relatives was a common practice in some ancient societies. It's too bad he didn't remember the study, because maybe he would have noticed the high proportions of farmer ancestry in this early Neolithic individuals, all of them females, and only one carrying a haplogroup strongly associated with Mesolithic hunter-gatherers. He maybe would have realized that this wasn't looking good. The Neolithic transition in Europe was characterized by a massive migration from Anatolia, but apparently Dan didn't know this, because otherwise he wouldn't have used a paper on this topic. More studies including new samples have been published since 2011, and there's really no reason to think the process was sex-biased, and we know the Anatolian haplogroups and admixture are not the same found in local hunter-gatherers. It was a bit different in the Balkans, with a local hunter-gatherer population that expanded before the arrival of farmers, already sharing ancestry with them. These hunter-gatherers, in a few cases, carry mitochondrial DNA that it's more associated with Anatolian Neolithic farmers, and these haplogroups have been reported in Mesolithic hunter-gatherers from Western Russia and the Baltic as well, groups that normally have more ancient North Eurasian admixture, which is also present in Mesolithic individuals from the Balkans, and pretty much absent in Western hunter-gatherers. So in this context, the low presence of these haplogroups was probably the result of population movements prior to 15,000 years ago. Either bidirectional gene flow between populations ancestral to southeastern Europeans of the early Holocene and those ancestral to Anatolians of the late glacial, or maybe a genetic influx from the former into the Near East. Since the early Neolithic, farmer ancestry became dominant in southeastern Europe, and we can see that there was a dramatic increase of the associated maternal haplogroups in Greece, with changes until the end of the Neolithic. In Hungary, farmer maternal haplogroups are dominant in the Neolithic samples discussed in this study, and most of them have less than 10% hunter-gatherer admixture, but one in particular has approximately 80% of this ancestry, even though it carries a very old variant of maternal haplogroup K. This study also includes results of individuals belonging to the linear pottery culture in Germany, showing a clear genetic turnover, but we're going to check another paper covering samples of the same culture, illustrating evidence of early contact between farmers and hunter-gatherers. The low coverage in this one could indicate the result is not as precise as in other cases, but it's interesting to see the tools and methods used by geneticists can still detect the main ancestral components in situations like this, and seems likely that this individual had quite high hunter-gatherer ancestry. They were not successful trying to determine the paternal haplogroups of the first two individuals, but you can clearly see this was not a problem to estimate their admixture proportions. Low levels of contamination were detected in the first individual, not finding signs of ancient DNA damage when checking his paternal haplogroup. The third individual shows no signs of hunter-gatherer admixture, which is great to see what an Anatolian farmer haplogroup profile looks like. Note it doesn't mean there are no other possible haplogroups, it's just an example, but same as we've seen evidence of admixture between both groups, others preferred to avoid any contact. In Brittany, for example, this paper discussed late Mesolithic hunter-gatherers who probably knew the Neolithic farmers existed, but they didn't mix with them, since their admixture and haplogroups are the typical found in Western hunter-gatherers. It's less certain in this case, because we don't know for sure if they were aware of the farmers, but it's important to consider that, for some time, maybe no admixture happens between locals and newcomers. Either because they don't want to, or because one of the groups is not interested in certain regions. The latter explains why in Europe, until the early Bronze Age, there was a clear east-west genetic division, described in this study as a Great Divide, finally ending with the expansion of Yamnaya steppe pastoralists from the Pontic-Caspian steppe. In this case, there are good reasons to think that the initial migration was male-biased, even though the number of women possibly increased in subsequent pulses, and high proportions of this ancestry were present in most, if not all, Western European regions in less than 1,000 years. It's certainly peculiar how the ancestral components are inherited when foreign men admix with local women, and seems clear there was no sex bias during the Neolithic, although this is more debated in regards to the steppe genetic impact. This study on a collective burial from 2500 BC and a Bell Beaker individual from 2300 BC, located in the Paris Basin, France, covers the topic in detail. It describes a mating bias of steppe men occasionally admixing with Neolithic women, whereas women of steppe ancestry mated less frequently with Neolithic farmer men. And here we can see which ones have an excess of Neolithic farmer and steppe-related ancestry on the X chromosome, and compare with the autosomes. It's not a new concept, and definitely not difficult to imagine, that if the migrants of the lost civilization were almost exclusively men, local ancestry would be more conserved in the female gene pool, while the opposite would be true for non-local ancestry. But that is one thing, and claiming it cuts the odds in half when it comes to finding evidence, or that every time a daughter is born it's game over, is another.
But I'm no geneticist, so I'll let them tell you how it goes.
Dan didn't understand the basics. So far, he just highlighted a sentence, explained very little of what geneticists do, drew inaccurate conclusions, and the details of the topics he chose not only are not representative of a male-biased genetic contribution, they don't even match his weird lost advanced civilization at all. Next, he discussed another paper.
In a paper titled Stable Population Structure in Europe Since the Iron Age Despite High Mobility, they collected the entire genome from 204 individuals, all long deceased. These were archaeological specimens, and they were taken from 18 different countries. They then took the specimens and compared their DNA to the local archaeological DNA to determine what may have been the product of migration and what was more sedentary. They found that 7 to 11% were outliers, that they had DNA that wasn't expected for that region and implied travel occurred. And what was the result of all that painstaking data collection? Well, here's the first sentence of their summary. In summary, we observed largely stable spatial population structure across Western Eurasia and high mobility of people evidenced by local genetic heterogeneity and cross-regional connections. These two observations are seemingly incompatible with each other under standard population genetics assumptions. So with all that effort, they had a margin of error of 1/3, 7 to 11%, and found stuff that makes them try to explain their seemingly nonsensical findings.
I'm afraid Dan didn't fully understand what the paper is saying. The 7 to 11% outliers basically refers to individuals who carried high proportions of non-local ancestry. He describes it as ancestry not expected for the region, implying travel occurred, and the paper, instead of not expected, says it's uncommon in the region. The difference is very small, but certainly not the same, and note Dan also refers to this 7 to 11% figures as a margin of error, which is not the case. The authors identified individuals whose ancestry was not the typical in the area, but in some of them it wasn't clear where the peculiar signal came from. That doesn't change, though, that the individuals in question are still outliers. 7% with source, and the rest without source. So what he said about the margin of error is simply made up. We can definitely expect outliers since the European Iron Age, some with ancestry not sampled elsewhere, and others carrying a mix of uncommon ancestries in the region and context they were found. So it's not really surprising they were not able to connect all of them to a putative source in a different region. On the other hand, the strict criterion for outlier consideration applied in this study implies that the percentage of samples with source should be regarded as a conservative estimate, as the authors were aware of other individuals that were not local.
They blamed their simulation on the error, and well, they listed as the first possible issue anyway. They used a simulation. They had 204 data points. They tested it against a solid baseline that they knew of the local populations and stuff, and they still couldn't discern crap.
They effectively identified the outliers and then investigated how the remarkable amount of heterogeneity and mobility impacted population structure over the last 3,000 years, which remained relatively stable according to PCA results. This might seem surprising at first glance, but it's normal to draw conclusions once the research is complete, and in this case, the authors proposed a very reasonable hypothesis. They remained cautious, and Dan discussed similar scientific reports in his videos. I have to say, though, that I've read terrible things about this journal, so maybe there are problems that I'm not knowledgeable enough to evaluate, but Dan's review can't be taken seriously.
All right, and this isn't some isolated example. This is extremely common.
By the way, a crucial detail is mentioned right at the beginning of the abstract, but seems like Dan didn't think about doing more research before concluding what's extremely common and what's not. He could have read numerous studies and seen the huge amount of data and results replicated multiple times, but anyways, the message is the following. Modern Europeans are not the same as ancient Europeans, and this is particularly obvious in individuals that predate the Bronze Age. You can learn more in my video on the origins of Europeans, linked in the video description. Let's now check the last study before discussing the part Mr. Dan the DeDunker still considers to be accurate.
When they would look for the Sea Peoples, for example. In Ancient DNA sheds light on the genetic origins of early Iron Age Philistines, we read, "Here we report the genome-wide data from the human remains excavated at the ancient seaport of Ashkelon, forming a genetic time series encompassing the Bronze Age to Iron Age transition. We find that all three Ashkelon populations derive most of their ancestry from the local Levantine G pool. The early Iron Age population was distinct in its high genetic affinity to European-derived populations and in the high variation of that affinity, suggesting that a gene flow from the European-related gene pool entered Ashkelon at either end of the Bronze Age or at the beginning of the Iron Age. Of the available contemporaneous populations, we model the Southern European gene pool as the best proxy for this incoming gene flow. Last, we observed that the excess European affinity of the early Iron Age individuals does not persist in later Iron Age population, suggesting that it had a limited genetic impact on the long-term population of the structure of the people in Ashkelon. Guessing at populations, hoping that their model is correct, and at the end of the day they admit this short-lived mingling doesn't even barely show up in the modern record. It's nothing in the modern day. It's non-existent. So this is an example of the troubles that we have when they start looking for genetic information that they really don't know the beginnings of. They have a hell of a time when they're trying to pin this stuff down. And we don't know Atlantean DNA, right? So this problem is definitely going to apply.
The Sea People seems to be the most similar example to the lost civilization proposed by Dan. I guess because they were seafarers, we don't know their DNA for sure, and let's assume they often traveled in small groups. Why not? The funny thing is, they might have found they came from Southern Europe, because some of the early Iron Age samples from Ashkelon show evidence of this admixture. So at least that's a starting point, and it shows DNA doesn't have to survive in the modern population for us to know it was there. The problem is that people with decent seafaring skills were very common in the Mediterranean at that time, which makes very difficult to discern who was part of the Sea Peoples and who wasn't. The master seafarers of the lost advanced civilization supposedly reached distant continents, something that it's not typical of the Ice Age or the Younger Dryas period. So once we identify samples with ancestry originating very far from the archaeological site and conclude sea travel was required, the implications become obvious. But finding Southern European ancestry in the Levant, or vice versa, is not particularly surprising during the Iron Age, so I'm afraid any attempt to extrapolate from this, even if more closely aligned with the kind of civilization Dan proposes, is doomed to failure. And well, modern genomes can definitely tell us lots of things, but I don't see how the genetic evidence for Dan's weird lost advanced civilization could be found without ancient DNA.
We can look for genetic outliers, markers that say travel between regions that historically are believed to have been separate actually occurred, but we can't go looking for Atlantean haplogroups because we don't know what they are or if they ever existed at all.
This clip is even more absurd than what he claimed about the implications of a male-biased migration. On one hand, Dan acknowledges we can identify the genetic outliers after saying all those things about the paper covering this, but then he says the same does not apply to the Atlantean haplogroups because we don't know which ones they carried. The ancestry outliers would be labeled as such for having distinct admixture proportions, thus not clustering with the local population, and remember we can know that without even being successful trying to test their haplogroups. So Dan didn't know he already admitted, before even presenting his smoking gun evidence, that we can definitely catch the Atlanteans if they really existed, and know their haplogroups if these can be successfully tested. By the way, we also don't know the ancestral components they supposedly carried, so if we start finding them scattered all over the world, we would need to compare their genomes against the available ancient and modern datasets to see who is more similar. That's how sooner or later, or with luck, we would know where they originally came from, or at least have an approximate idea, because the problem are not the tools, methods, or whatever aspect geneticists take into account, as Dan pretended, but having the appropriate samples that match. If we already have them, they would be associated with the lost advanced civilization, and the source wouldn't be a mystery anymore. And now, let's examine what Mr. Dan the DeDunker has not retracted yet.
But assuming that they did exist, we wouldn't find them anyway, not the way the work is currently done now. Now, I know you're going to have a hard time believing me, so if you're one of those people that really puts DNA up on a pedestal, you might want to sit down here because this is going to sting a little bit. As discussed previously, DNA is crap for determining dates, so we actually have a serious problem. You want to sequence DNA from Australian Aboriginal people, for example, and you have to account for the European DNA that was mixed in since the settlement of Australia by the English. But they have almost no way of determining when the DNA was actually introduced to the lineage, and to solve this problem, they've got a very inelegant solution. if there is anomalous DNA from another population that they know has been in that area at all, they just exclude that DNA. Now, I know a lot of you right now think that I'm just lying, right? That this is just complete bullshit. But now, here, bear me out here. The second most popular, second most common haplogroup in the Native American populations is an Old World one. It comes from the Old World. It's not a New World haplogroup. It's the second most common. So how do they deal with that? Just like I told you. Listen. All individuals that did not belong to haplogroups Q and C were excluded from the haplotype dataset because these haplotypes are likely the result of non-native admixture. Note the multiple citations. This is not some novel method employed by one or two geneticists. This is the way it's done. The historical framework created by old school academia is used to inform the hard data that they get from the genetic studies. And in the case of Native Americans, for example, they assume that any Old World DNA came from post-1492, and therefore if they exclude it when they're looking into like things that happened before 1492, they assume that this DNA had nothing to do with that. So if our hypothetical Atlantean DNA had Old World haplotypes in it, and they had settled in America, those haplotypes, when they are found in the Native American population, they will be discarded. They will be ignored. They will be thrown out as post-1492 haplotypes. Period. End of story.
Dan presented a study published in 2008 as the smoking gun evidence confirming that geneticists always exclude a possibility based on mere assumptions. But as already stated, he moved the goalposts months later by claiming his main point was about how the DNA is tested in the Americas, which was very dishonest of him and still completely wrong. I will show you where that study is coming from, but as many of you probably imagine, more research was published after this paper, and it turns out that West Eurasian ancestry much older than 1492 has already been demonstrated to be present in Native Americans. In 2014, this study found that the Kurishana, who along with the Surui are very representative of Native American ancestry, carry significant West Eurasian admixture, but coming in this case from an Upper Paleolithic Siberian source, represented by the MA1 sample and other similar ones. Actually, both modern West Eurasians and Native Americans share this ancestry, that we know it existed thanks to ancient DNA, as early as 24,000 years ago, being the samples in question intermediate between modern West Eurasians and Native Americans, but distant from East Asians. This applies at least to the MA1 and AG3 samples, the latter being more recent and very similar, but not exactly to the AG2 sample, which is dated 1,000 years older than AG3 and found to be significantly contaminated. The findings were published more than 10 years ago and are still relevant today. You can see here Dr. Yosif Lazaridis, a living legend in the field of archaeogenetics, showing he still had a good opinion about the paper in 2024. He and other authors also published a study in 2014 validating the previous work, and in 2017, another paper reported weird results indicating that the gene flow between MA1 and the Kurishana occurred the other way around. However, this was not what the scientists were mainly investigating, and it's quite apparent that they didn't believe these particular results made sense, as explained in this paragraph. Anyways, Dan wasn't even aware of this, and if that were the case, he probably would have used the results out of context to avoid my critique, as his reaction when I showed him the 2014 paper clearly indicates. I thought Mr. Dan the DeDunker deleted the post announcing his DNA video, because I really wasn't able to find the interactions in question, but they still exist, so we'll be checking some of those comments, and others posted in another thread started by one of his fans. This dude believed what he was saying, and decided to share the content, so I basically replied to tell him it was not correct, and Dan immediately came to stop me. The authors of the study took care to ensure that the ancient North Eurasian signal was not mistaken for recent European admixture, so the possibility was not excluded based on assumptions, but Dan desperately insisted that this quote solved his problem, and that the study I shared supported his point. Note that it's literally impossible that research reporting West Eurasian admixture far predating 1492 supports his point, since they shouldn't be finding something he said cannot be found. The source of the ancestry was not in Europe or West Eurasia, but let's not forget that the ancient North Eurasian admixture has strong West Eurasian affinities, and it's part of the modern European gene pool, being quite high among Northern Europeans. Dan was constantly misreading and misinterpreting the content, while still pretending that the quote he highlighted was crucial, but I missed there was a paper linked, so I acknowledged it and, after trying to guess, proceeded to check. This paper, published in 2012, identified individuals with recent European and sub-Saharan admixture, but it wasn't a guess or a mere assumption. What he calls contamination is not contamination. The samples were admixed, not contaminated, which is something that only applies when exogenous DNA is detected in a sample. The methodology is not the same as the 2008 study that Dan used to prove he's right. And even if it's true that most analyses were performed on a masked data set that basically subtract the expected non-native contributions, this doesn't mean what Dan pretends at all, but there's no way he could know without taking the time to read and understand the Reich et al. 2012 paper. There was a number of populations for which they didn't have access to unadmixed samples, so the masking procedure was essentially used to learn about their history. With local ancestry inference software, they identified segments without evidence of recent admixture, since yes, local ancestry segments can be used to infer various things, including the timing of admixture. There was a paper published 1 year before this one doing exactly that, so they clearly had the possibility to do this back in 2012, and even though they didn't provide dates of admixture in this case, it's quite apparent they made sure to retain segments with an extremely high nominal probability of being native, meaning those excluded were of recent origin. The authors also state that masking produces inferences about history that are consistent with those based on unadmixed samples, and thanks to this, there's a much easier way for us to discern if the ancestry is recent or not, which is checking the results listed in the supplementary material. We can see populations like the Cariciana or the Surui are 100% native, but these are not the only ones. Other indigenous groups from Brazil, Argentina, Colombia, and Costa Rica show no signs of non-native admixture, but the populations that really matter to us are the ones including both admixed and unadmixed samples. It's precisely when we check the individual results that the whole thing really looks terrible for Dan. Here we have some native individuals from Canada, most of them with European admixture, but we can clearly see 2 of them lack it, and likely the other one with a very low percentage of European ancestry, which is similar to what we find in the East Greenland samples and other ethnic groups. The same individuals were analyzed in 2019, and again, 3 of them did not have any European or sub-Saharan admixture, not testing the East Greenland samples in this case, but more populations were tested here as well, and we keep finding admixed and unadmixed individuals in the different ethnic groups. Also, whole genome sequence data was analyzed in 2024, and there we can see 2 different Chippewayan individuals that, unlike other samples, don't deviate towards Europeans, so I think that's enough to figure out what's going on here. The possibility that the European admixture could be 12,000 years old, being so widespread among people who identify as Native American, but still absent in some populations and individuals today, is extremely hard to swallow. Nothing can explain how some individuals remained isolated within their own ethnic groups for more than 12,000 years. It's basically inconsistent to find some with 100% native ancestry, while others are more than 30 or even 40% European. So the only plausible explanation is that the European admixture must be recent. Otherwise, all the individuals included in the different ethnic groups would be far more homogeneous. Simple as that. Add to this the much older West Eurasian signal of ancient North Eurasian origin, and there is no doubt that Dan's stuff doesn't hold water. I recommend reading what is said in these papers multiple times, because it's easy to miss or misinterpret some things, and I have no problem admitting that when I discussed the 2012 study with Dan, my comments pointed in the right direction, but were still poor. Also, I had the impression that Dan didn't believe it's possible to tell the difference between one DNA signal and another, even though the 2014 paper clearly says they take recent European admixture into account, but conclude that's not the origin of the signal. He then said he had no problem with geneticists being able to tell the difference, so he essentially took issue with the DNA he claims they exclude based on assumptions. It's pretty clear by now that this is not the case, as there are ways to tell if the ancestry found in modern individuals is recent or not, but there's still more to discuss about haplogroups and ancient DNA. As one of the papers cited by Dan indicated, ancient Europeans are not the same as modern Europeans, and this is particularly obvious in samples that predate the Bronze Age. Nobody has this admixture proportions today. Only a few Sardinians are almost identical to some early European farmers, and most, if not all, still carry some steppe ancestry, even if at very low levels. Before the Mesolithic, the inhabitants of Europe were also unique genetically speaking, and there is evidence of admixture between Upper Paleolithic and Mesolithic hunter-gatherers. In this global PCA, you can also see very old samples found in Europe, which also includes ancient North Eurasians and other references, and it's easy to notice they don't cluster with modern West Eurasians, even though some deviate more towards them than others. The reason I explain this is to clarify that when testing ancient DNA, there's no way geneticists would dismiss high proportions of certain ancestries as the result of modern contamination, primarily because some genetic signals either no longer exist today or are found in much lower proportions. But to be fair, that doesn't really show how they deal with the problem, so I'll talk about it later. Dan's line of reasoning could only apply to possible migrations from the Bronze Age onwards, that for the most part, we have no evidence they occurred, or to Vikings admixing with Native Americans, but their genetic contribution was either negligible or non-existent. Low frequencies of haplogroups I1 and R1a among people who identify as Native American suggest the influence was not significant, and the European admixture found in Pacific Northwest Native American groups has been estimated to be very recent, even more so than what has been reported in mestizo populations across the Americas. Testing more ethnic groups may show different results in some cases, particularly those who identify as Native, but so far, the findings are inconsistent with Vikings spreading a significant amount of European ancestry. Modern Europeans, whether from the north or the south, simply did not exist during the Ice Age according to published research, so Dan was trying to make a big deal about something that has no impact whatsoever when it comes to the alleged lost advanced civilization. The 2014 research wanted to investigate Native American ancestry, but as noted in the 2012 paper that Dan desperately brought to the conversation, recently admixed individuals are problematic for that purpose, so scientists can either exclude the samples or create a mass data set. The DeDunker can keep crying about it, but there is no flaw or sign of dishonesty here. The DNA in question is recent, and the papers simply prove him wrong. We're now going to see how much of the 2008 study was based on assumptions and learn about the most common branch of haplogroup R1b found in Native Americans. Some might think an ancestral component could be gone after 12,000 years, even though that would certainly apply to all the samples, yet we find important differences among individuals in Native American groups, ranging from 0 to more than 45% European admixture. We also have the much older ancient North Eurasian admixture reported in Native Americans, which is in part West Eurasian, but we're going to ignore this now and assume there was another very old West Eurasian signal originating in Europe that, unlike this one, it's no longer present in the autosomes. So we can then consider the possibility that haplogroup R1b may be the only thing left after more than 12,000 years in the Americas. I wasn't familiar with the old DNA papers, so when I checked the 2008 study for the first time without reading in detail, I only saw the samples belong to haplogroup R, and it's definitely not impossible that this one entered the Americas a long time ago, since the Upper Paleolithic MA1 sample belongs to this haplogroup. They performed short tandem repeat analysis, and observed a strong gradient of haplogroup R from northeastern to southwestern North America, which in their view reflects earlier contact with Europeans in northeastern North America, and thus provides a longer period of time for admixture to occur. This corroborated the findings of previous studies, and it's also noted that Y chromosome analysis shows Native Americans harbor a high frequency of alleles of European ancestry, but this 2006 paper is more detailed when it comes to the evidence suggesting that haplogroup R came with European colonists after 1492. In contrast to what STR markers revealed when they tested the other haplogroups, the RM173 network showed no clear-cut patterns in Eastern North American groups. Haplotypes in this network do not cluster by population, culture area, language family, or geography, which is consistent with the idea that this haplogroup represents post-1492 European admixture in Eastern North America, rather than a founding Native American lineage. Also, the most common RM173 haplotype in Eastern North America is the most common in Europe, but rare in Asia. The 62 other RM173 haplotypes found in Eastern North America appear to be rare or absent in Asia, whereas 43 of them are common in Europe and 14 others are one mutational step away from known European haplotypes. 5 haplotypes are 4 to 8 mutational steps away from European haplotypes, but they probably also stem from recent European admixture. The median joining network suggests that they derive from separate European haplotypes, whereas one would expect them to cluster together in a distinct clade if they represented a founding lineage from Asia. Thus, the author's interpretation was that few, if any RM173 haplotypes in Eastern North America are likely derived from founding lineages of the Americas. Dan would probably take issue with the fact that the authors only mention Asia as the source of non-recent lineages, but it still doesn't make any difference, because if it were true that haplogroup RM173 was very ancient, the patterns would likely look different no matter where it came from. The other paper cited in the 2008 study is also very interesting and uses unambiguous SNP characterization, showing that the vast majority of individuals who identify as Native American and carry haplogroup R specifically belong to RM269. A similar pattern can also be observed in both African and Hispanic Americans, who have very low levels of the other West Eurasian haplogroups, all of which are expected to have arrived alongside haplogroup RM269.
Look at the margin of error for DNA studies, especially when you start going back 5 to 10,000 years, man. They really, really don't know. They can't use DNA to pull out a timeline and say this people were here 5,000 years ago. They were here between 6,000 and 16,000 years ago is more like the kind of numbers you'll get into. But again, I don't want to make this too data heavy. You can go look into that yourself. But if you're going to be all like, oh, we have no DNA for that, miss me with that shit because you ain't done no fucking research.
It's not clear what Dan was referring to when he mentioned the margins of error, but it was probably something he read about haplogroups, which cannot be associated with groups of people without additional testing, and it's important to note that haplogroup RM269 is among the ones we know the most about. Back in 2010, some scientists already pointed out it's quite young, while Morelli et al., for example, proposed an origin before the last glacial maximum, but more recent studies and the current ancient DNA evidence strongly indicate that it's nowhere near as old. The time to most recent common ancestor is estimated to be around 6,500 years before present, but we can see from this tree that the haplogroup was formed 13,300 years ago, which might lead some people to think a 12,000-year-old expansion is plausible. Yfull.com has an FAQ section where it's possible to learn about the methodology, but this particular detail is confusing, because what we're looking at here isn't the divergence point, but the TMRCA of its parent haplogroup. In other words, the mutation did not occur 13,300 years ago, but sometime between 13,300 and 6,500 years ago, and 2 more factors should be taken into account. How long it took for a significant number of people to share the same marker, and the evidence indicating when it began to spread more widely. The former doesn't happen overnight, though it's certainly hard to pin down, and at a local level, we can expect it to occur sometime between the emergence of the actual mutation and the accepted TMRCA. So even without precise dates for every single event, we can roughly tell that haplogroup RM269 is not old enough to have reached the Americas during the Younger Dryas or the Ice Age. But what really makes it very difficult to believe is the abundance of ancient DNA samples from Europe. So let's see what we have. This is the Ancient DNA Explorer website, where thousands of samples are listed, and you can easily tell that Europe has been the most thoroughly investigated region. We're going to use the site as a starting point to figure out which samples could be the oldest known to carry haplogroup RM269, but verification is certainly needed, since there are some errors and important details are often missing. So let's type the relevant haplogroup and check. This sample is very old, for example, but I checked the supplementary materials of the original study, and it was excluded due to contamination and low coverage, so it's not reliable. Another one I found interesting was this sample from Romania, but in the section that contains details for all the samples from the study, we can see that it's a female individual listed as unpublished, which suggests that the reported paternal haplogroup must be due to contamination. Then we have this one that it's too old and was excluded because only haplogroup A could be determined, which is not informative and implies that what we see here is not correct. And we have a similar problem with this one from Iraq, because in reality this sample could only be assigned to haplogroup F. So let's now focus on the ones that look more promising. This one from Latvia is not incorrect according to the original publication, and there are others not included on the website that appear to be quite old as well, although I think RM269 is not 100% confirmed based on how the results are listed in the table. But even if we take it as fact, it's important to note that the Younger Dryas ended more than 2,000 years before the oldest one. Next, this one is controversial. It's a Chalcolithic outlier with steppe ancestry from Bulgaria, which the original study assigned to haplogroup R, but in the Southern Arc paper the authors apparently believe it belongs to RM269, and two more studies list it as belonging to haplogroup F. So what we see in the website, although possible, is dubious, but the steppe ancestry is definitely very interesting in this case. Finally, this sample seems to belong to RP297, which is basically the ancestor of RM269, and there is also a 7,500-year-old individual assigned to this haplogroup in the study titled The Genetic Origin of the Indo-Europeans, published in 2025. So there was something going on in Eastern Europe and the Baltic prior to 4,500 BC, and it's very likely that some of these samples belong to RM269, but they're still too young to take a 12,000-year-old expansion seriously. We also have a pretty good understanding of what was happening in Western Europe before the Chalcolithic period and early Bronze Age, and haplogroup RM269 was virtually absent. The spread of this haplogroup is clearly associated with the expansion of Yamnaya steppe pastoralists approximately 5,300 years ago, and the process continued with subsequent cultures carrying high levels of steppe ancestry. To my knowledge, not a single Mesolithic or Neolithic individual from Western Europe has been reliably assigned or predicted to belong to RM269, but even if I'm missing something or one is eventually found, the point still stands. Its presence was insignificant, and no population turnover associated with it has been observed during these periods, unlike what's obvious in the early Bronze Age, or in the Americas after 1492. So not only is the time depth required to match the lost advanced civilization very hard to believe, it also doesn't make any sense that haplogroup RM269 expanded to the Americas during the Younger Dryas or the Ice Age without having disrupted a significant portion of the European territory, and nothing suggests an entry from Northeast Asia. Dan should explain to the audience how he isn't biased, and how he can speak so confidently on certain things, because even though he has access to a ton of information, He often ends up spreading falsehoods while focusing on extremely unlikely possibilities, and I'm being generous. There is a more likely route from Northeast Asia that could explain the hypothetically very old RM-173 branches in the Americas, which makes sense considering the geographical location where the MA1 sample was found, and also the presence of ancient North Eurasian admixture in Native Americans, characterized by strong West Eurasian affinities. However, all the archaeological specimens tested in the Americas so far resemble Native Americans or Siberian populations, not Europeans or modern West Eurasians as a whole, and not a single one belongs to Haplogroup R, let alone R1b. So to be clear, they didn't know a lot of things when the old papers were published, but there were still important pieces of evidence to support their conclusions, which have been further strengthened by subsequent studies.
DNA is happening so fast that I made a mistake once in a video because I just went with what I'd learned 15 years ago and 10 years ago, and that's RONG wrong now. But with the DNA, we are able to suss certain things. But if you start from a ground point, like say for instance, you assume that this is the DNA of this culture, right? And then you start looking for other ways that they bred out into other places and stuff. You're starting from an assumption that that is the way that these people were. They can only go back so far with what they test. The margin of error gets wider and wider, and the world's been in contact for a long time. Like one of the things that is true, it's not as inaccurate now as it was years ago, but one of the things I put in that video that was true was when they test say they test a Native American today and they find European DNA, they assume that that's post-Columbian DNA for obvious reasons. But that does also say that if they were to test Native American and find pre-Columbian DNA that was the old world, they would assume that it was not pre-Columbian, that they would assume that it was post-Columbian. So when they look at these things, they attach, there's already a framework, already a worldview attached to it. And so the margin of error on some of these things can be thousands of years. But they'll slide it into where it fits because this is where it fits. And that's not to pick on them about that. There are places I will pick on them all day long, and I won't pick on them about this because this is a new science, and they're trying to reconcile it with what we already know.
The post-1492 European admixture in modern Native Americans is based on genetic evidence, not assumptions as Dan claims. And it's too bad he already misinformed thousands of people in this podcast repeating the same stuff, while omitting, of course, the embarrassing story behind his DNA video and the retraction. His claim is 100% false, but even if we ignore the fact that it doesn't stand on its own, he avoids mentioning whether what he calls a mistake actually matters to our understanding of the topic or is entirely irrelevant. And among other things, Dan completely underestimated what we have learned thanks to ancient DNA, and apparently keeps insisting on it. But the reality is there's a huge body of evidence that supports a recent origin for the European DNA found in Native Americans, which comes from a large data set including both modern and ancient individuals and multiple tests. Many probably find Mr. Dan the DeDunker entertaining, but his tendency to oversimplify complex topics and distort the evidence, combined with frequent major errors and outright lies, definitely says a lot about the level of scientific literacy he often brags about.
We could have smoking gun evidence, piles of it, of out-of-place DNA that proves the existence of Atlantis, but they are throwing it out because they assume that it came over post-1492. And this is reality. Again, you might think that I'm full of it, but you can go dig into it a bunch. Even Wikipedia knows this is the case. And this is a rule worldwide, but it's easiest to show in the Americas, since that area was presumably isolated for so long.
We all now know that this is not true, and Wikipedia knows this as well. It was actually Dan the one who had the answer in front of him, but couldn't, or didn't want to see it. He did not understand the implications of this, and missed the studies cited in both the smoking gun evidence paper and the Wikipedia article. He also failed to connect the dots when reading the Iron Age study that was giving him a very important clue. The Atlantean DNA, assuming it came from Europe, is not possible to be identical to what is found in modern European populations, and unique mutations would most likely be present in haplogroups found in modern Native Americans after more than 12,000 years, no matter if they came from Asia or Europe. And let's be honest, RM269 is not a good candidate at all, but that doesn't mean it's not worth investigating other possible interactions that are not as old, yet still predate 1492, like the Vikings, for example. Ancient DNA is ideal for that, but here we are once again, correcting Dan for extrapolating from something in a very unfair way. For context, he was commenting on a press article that discussed the lack of a clear genetic evidence for the Danish Viking occupation of England, which let's assume was quoting a 2015 paper accurately. It doesn't matter cause another study published in 2020 found this evidence, although it's true that pinpointing the exact percentage of Viking ancestry in the British population remains difficult, since Vikings and the British are very similar, and the same applies to the Anglo-Saxons. This is not just my opinion, it's clearly stated in the paper as well, which also indicates that the Vikings were not a homogeneous group. Viking DNA in modern admixed Native Americans, I guess it's not easy to estimate assuming some have it, basically cause many of the post-1492 Europeans probably carried some, but this problem wouldn't apply to the archaeological specimens, since unadmixed Native Americans and Vikings only shared very old ancient North Eurasian admixture. So pretty sure that if archaeologists find human remains in North America, date them to around 1,000 years ago, and the DNA analysis shows high proportions of Native American and Northern European ancestry, the results wouldn't be difficult to interpret. There's no reason to think this based on what we've seen regarding the Malagasy, as well as the interactions between Neolithic farmers and Mesolithic hunter-gatherers in Europe that were genetically much closer than Austronesians and Bantu peoples. Dan was either dishonest or ignorant regarding the challenges of examining certain possible interactions, and when it comes to his weird lost advanced civilization, ancient DNA is crucial, so the lack of samples will always be the biggest problem, but discerning the ancestral contribution from a specific group to other populations that are genetically very similar isn't something to worry about in this case, since the areas reached by the lost civilization were inhabited by very distant groups. The DeDunker was wrong about way more than he admitted, claimed things he did not even believe, doubled down, and if I hadn't posted on social media that I was making this video, he wouldn't have retracted anything. He then thought it was a good idea to lie in his retraction, and got exposed for that as well, so his decision only made the disaster worse.
But look into it a little bit. I'm not trying to make this video too data heavy, but there is anomalous DNA all over the world that proves different cultures came into contact with each other. And that's not even the right way of wording it. It's not different cultures. It's different people from different regions, different individual groups of people, but not different cultures per se.
There is anomalous DNA all over the world according to Dan, but he doesn't bother to define it. It seems like if the DNA is not local, it doesn't matter if we have a good explanation for it. Dan can just ignore it, dismiss it as an assumption, misrepresent whatever he needs to, and sell the viewers it's anomalous. We all can look at this picture without doing any research and draw inaccurate conclusions, but the subclades of mitochondrial DNA X reported in Native Americans are not found outside the Americas and do not derive from Old World lineages.
Genetics has become a very important field in understanding the movement of human populations. By comparing the DNA of modern human populations with DNA recovered from ancient humans, scientists can piece together the movement and development of their descendants. This has shed a lot of important light on our understanding of human migration and settlement in the Americas. If the Solutrean model is correct, people from Western Europe should have arrived in the Americas between 18,000 to 15,000 years ago. Assuming that any of them survived and passed on their genes to modern Native American descendants, there should be detectable genetic traces in ancient and modern humans. I do want to mention a quick caveat, and that is that when we look at indigenous North American genetic evidence, both modern and ancient, we are looking at small sample sizes that are probably not giving us a complete picture. It goes without saying that our understanding could change at any time with a new discovery. So with that out of the way, does the current genetic evidence show any ancient lineages that could have derived from a European source? Some proponents of the Solutrean hypothesis have pointed to the presence of Haplogroup X as a link between North American indigenous groups and peoples living in Europe. A haplogroup is a group of people that share specific genes that are derived from a common ancestor. Haplogroup X is found in indigenous North American populations and in modern European and North African populations. At first glance, this looks very compelling, and it's tempting to trace an imaginary line between North America and Europe and speculate that there was a migration. However, the genetic picture is actually more complicated. What many fail to realize is that the populations in Europe and North America belong to different clades or subgroups of Haplogroup X. Europeans with Haplogroup X lineage are in the X2b, X2d, and X2c clades, while Native Americans with Haplogroup X lineage are in the X2a and X2g clades. This is important because geneticists can see that those lineages in America do not descend from the lineages in Europe. Actually, neither of them are derivative from the other. A good way to think of this connection is as a cousin relationship rather than a parent-child relationship. These X2 clades all appear to have a common ancestor in Eurasia long ago, but where and when this split occurred is still not known for sure.
This was a great summary, but despite the fact the evidence is either absent or too weak to take it seriously, there are still studies pushing the Solutrean hypothesis in 2025. In this case, for example, only a few papers are cited to support this particular claim, which, as we're about to see, is not very accurate. The paper states that the X2J lineage is closely related to the American X2A, but such an oversimplification is quite misleading. Both lineages emerged from the X2AJ clade, but another study published in 2012 that the authors of the recent paper do not cite points to considerable independent evolution, and it's actually the first one to check in order to understand what's going on here. We learn that both lineages show the same mutation at position 12,397, and in their maximum parsimony tree, it seems to be relevant to define X2AJ, but they note X2J is significantly younger than X2A, which indicates the mutation might be a recurrence, so rather than being inherited from a common ancestor, it likely occurred independently in both lineages. This is known as homoplasy, and it's well documented in mitochondrial DNA studies to affect both coding and non-coding regions, and it's relevant to differentiate IBD and IBS segments as well. Now it's easier for us to revise the 2003 paper, which does not explicitly refer to how connected X2J and X2A are, but reports an X2 sequence from Iran, sharing the same mutation with the American X2A at position 12,397. However, the authors remark that the same has been observed in two different phylogenetic contexts, and conclude recurrence is the most plausible explanation. So unlike what the 2025 paper pretends, it doesn't support the Solutrean hypothesis or the possibility of gene flow from America to Europe. No one questions that X2J and X2A are sister clades united by the X2AJ branch. What's debated is how connected they really are, and the more connected, obviously the better for the proponents of the Solutrean hypothesis, because they need some evidence to prove that the first inhabitants of the Americas came from Europe across the Atlantic. X2J is associated with Europe in the 2025 paper, even though today is more common in North Africa, but it is very possible that it was brought there from Europe a long time ago, since a couple of early Bronze Age samples from North Italy belong to that subclade. Anyways, both Europe and North Africa are geographically acceptable for those who believe in the Solutrean hypothesis, because in their logic, it would make sense that the Solutreans also traveled from Iberia to North Africa, but if position 12,397 were reliable, which some forget to mention it's not, a migration from Europe to the Americas by sea wouldn't seem so unrealistic. We know nothing about the distribution of X2AJ, X2A and X2G are restricted to the Americas, and X2J is extremely rare, even in North Africa. But for the proponents of the Solutrean hypothesis, the lack of lineage is ancestral to X2A in Siberia, the mutation we've discussed, and the fact that the oldest X2A lineages carried by modern people are found in northeastern Canada should be enough to take it seriously. Unfortunately for them, the last argument was torn to shreds once Kennewick Man was tested, as the individual carries the most basal X2A lineage found so far, meaning that approximately 8,500 years ago, the lineage was present in the Pacific Northwest, literally right at the opposite side. So following the same logic, the finding supports a migration across the Bering Strait and not the Solutrean hypothesis. There is also no paternal haplogroup that can be used to support it, and the autosomal DNA evidence is completely lacking, unlike what we've seen regarding recent European admixture or the ancient North Eurasians.
It's imagination, it's a dream. Let them to live in dream.
Everyone's free to not discard the Solutrean hypothesis, of course, but if we're honest, there's a real need for new evidence to back it up. Moving on, it's important to note that in the US, a lot of indigenous groups disappeared, and the remaining Native American tribes are considered sovereign nations, meaning DNA studies, including ancient DNA, are conducted on a case-by-case basis, so comprehensive datasets aren't always available. Also, many of the paternal haplogroups found in North American natives could have been tested in more detail, even though the evidence supporting recent European admixture is still overwhelming. When it comes to Mesoamerica and South America, though, the information in question is easier to find, and here's an example of what they reported in 4 Amerindian populations from Ecuador that have minor presence of West Eurasian and African haplogroups. Some individuals in these groups carry subclades that are even younger than RM269, which is no surprise, but it's even clearer in the next study published in 2024, where we find many elements discussed in this video. In the paper called Ancient Genomes Reveal Insights into Ritual Life at Chichen Itza, we have a lot of ancient individuals that we can compare with the present-day people who live in the region, and there is absence of European Y chromosomes among the ancient individuals, in contrast to the modern ones, who not only carry R1b subclades, but also other haplogroups that are expected to be found in post-1492 Iberians. The estimated European admixture for the modern samples is 7%, and considering that no mitochondrial DNA from Europe has been detected, this reflects a strong sex bias in the admixture process during and after the colonial period, as has been previously described in other Latin American populations. And it's super interesting that 18 of these modern individuals show no indications of non-Indigenous American genetic contributions, which is basically the same we've seen in other Native American populations, further confirming a recent origin for the European admixture. There is one sample among the ancient individuals showing some European admixture, but it doesn't mean what some of you think it means, and I'll cover it in a moment. By the way, I'd like to clarify that examples of haplogroup R1b not associated with recent migrations have been identified outside Europe, like the V88 subclade, that has nothing to do with RM269, but we can definitely include this one as well, as it has been reported in North Africa, for example, and was likely brought there during and after the steppe-related migrations in Europe. The Romans probably had something to do with this, but part of it could be much older, since pottery associated with the bell beaker culture has been found in North Africa, and admixture analysis also shows steppe-related ancestry in post-Neolithic archaeological specimens, like the Guanches, who before the Islamic invasions settled the Canary Islands from the Maghreb. Back to the settlement of Madagascar, there's a small West Eurasian component among the Malagasy that, for the most part, it's not attributed to recent European admixture. Post-1492 Europeans were definitely very disruptive in the Americas, but not even close in Madagascar, where it was difficult to establish colonies due to the hostility of the locals. The legacy of colonial rule is still undeniable, though, as French remains a common language that is widely used in business and government, but the West Eurasian ancestry is mostly attributed to Indian and Arab traders, who may have brought small amounts of it to Indonesia, which explains why there is minor presence of West Eurasian haplogroups there as well, including some shared with the Malagasy. This means the component either came from Indonesia due to some admixture prior to the occupation of Madagascar, or directly from the aforementioned sources. Also, let's not forget that according to Hancock, the lost advanced civilization was comparable to late 18th century Europe in terms of seafaring skills, so even if we assume they had very different technology, it's pretty safe to say that the evidence should be widely distributed, though not necessarily everywhere we look.
Nothing to see here. And now let me be very clear here. I love science. I'm big on science, and I'm not trying to say geneticists do their shit wrong. Not one bit. They should not change these controls. They should absolutely take on 1492, for example, was a big time in the changing of the admixture of the Native American freaking population.
It's no wonder Dan wants geneticists to do things in a certain way, but what he said is completely false. Both the initial claim that it's a global rule to assume recent admixture explains some of the DNA they find, and the later attempt to move the goalpost by pretending his main point only applies to the Americas, when in reality, he was using it as an example of how the work is supposedly done everywhere. It's beyond laughable, and the same applies to this post, because there is no doubt he didn't do his due diligence, and the premise that their models must account for hyperdiffusionism in order to find evidence for it is simply not true. Scientists don't necessarily need to consider something in order to find it. It can also unexpectedly appear in their results, and a good example to illustrate this is precisely one that Hancock himself popularized, which is curious that Dan didn't mention considering he pretends to be well-versed in Hancock's work.
Advances in DNA technology are making these kind of explorations much more possible. And what's been discovered is this very puzzling connection between the peoples of Australasia, particularly Papua New Guinea, and certain tribes deep in the Amazon rainforest. Now, this is a real mystery because the view, I'll come to that, what makes it even more of a mystery is further research in 2018 on members of these tribes and looking for ancient skeletal remains. They found ancient skeletal remains 10,400 years old in the Amazon, which bear that same signal. And this tells us that this Aboriginal connection reached South America very, very long ago, and researchers are agreed that it must have reached South America in the late Pleistocene, which is near the end of the last ice age. It's a very important study because it establishes that there's no recent connection here. This is a very ancient connection in South America. And it really creates a problem because the view of archaeology for decades, really a century, has been that all the peopling of the Americas happened this way. That they crossed the Bering Straits, which were a land bridge during the Ice Age. Sea level was lower then, and that they traveled through North America and eventually down into South America. The problem is that this genetic signal isn't found anywhere in North America. It's only found in South America and in Papua New Guinea and neighboring areas. And I talked to Eski Willesleb, who's one of the world's leading geneticists, whose recent work focuses on the peopling of the Americas. And I'll just read a bit of this email exchange. Willesleb told me that currently no one has a good explanation of the Austro-Melanesian signal. All that's put forward as possible explanations are purely speculative. What we do know is that it's present in some Native American groups, particularly from Brazil. We also know it has to be pre-Columbian. Amongst the possible explanations for the presence of this Australo-Melanesian DNA in the Amazon, Professor Willeslov offered in his email exchange with me the following. Someone holding this signal comes into the Americas not through Beringia, but crossing into South America across the ocean. Based purely on the genetic data, this is the most parsimonious explanation, but it does not make practical sense. So I asked Eske why
it doesn't make practical sense. Is he basing that on the archaeological consensus that our upper Paleolithic and early Holocene ancestors were incapable of undertaking long transoceanic voyages? And his reply, in regard to crossing the Pacific, I'm not saying it did not happen, but there is no evidence suggesting that humans were capable of such a journey until quite late in history, the Polynesian expansion. It's a possibility, and I'm open to the idea, but there's not much evidence supporting it except going for the most parsimonious solution of the genetic data.
It's a very interesting case, and it is no surprise that Hancock prefers to focus on the opinion of one scientist, but should be enough for Dan to realize that what he's claimed is very far from the truth. Geneticists definitely consider possibilities that Dan pretends they dismiss out of hand. And even those who don't agree with what Dr. Willerslev told Hancock were surprised with this discovery. There are 2 videos available featuring experts in the field discussing this, one published by Stefan Milo and the other by Flint Dibble, where you can easily verify the last statement. The population Y signal in some South American groups was quite unexpected, but there are arguments, of course, questioning the idea of a long-distance journey by sea. It's unclear why this deep relationship can be detected, but very interesting clues were found in the area of modern-day eastern China, basically thanks to the 40,000-year-old individual called Tianyuan man, a sample with affinities to some South American populations, stronger even in some cases than the observed for the Papuan and the Ongi, indicating that the group responsible for bringing the signal to the Americas was once widespread in Eastern Asia. It's a complex puzzle that requires more ancient DNA to be solved, and scientists are carefully looking into it, including the possibility that the connection may just be a false positive. However, the signal is most likely real, and its absence in the populations of North and Central America studied so far can be explained if it was introduced through the Pacific coastal route, although more recent evidence suggests it could also derive from genetic structure in the founding Native American population, and not multiple independent migrations into the Americas, as indicated by the sporadic identification of the population Y signal in ancient individuals with different ancestries, locations, and time periods across Brazil, according to this paper published in July 2023. What we know about the population Y signal isn't really as promising as Hancock would like, but he has the right to believe whatever he wants and pretend it supports his case. The truth is he can't prove anything, and all he has left is weak speculation that doesn't take into account the totality of the evidence. The DeDunker did not disappoint, and recently sided with Hancock when responding to Stefan Milo.
Look at the doublethink on display here. On the one hand, DNA is so good you could just do this, and this with it. On the other hand, if DNA supports the possibility of a civilization of Graham's liking, and the possibility, doesn't have to be supporting Graham, just a possibility. And now, well, maybe they came down through here, and maybe so many of them got wiped out here that they lost the signal, and maybe some of them interbred over there. It's tough to say for sure. That DNA is tricky stuff when Graham brings it to you. But when he doesn't bring it to you, where's the DNA? This is why Graham prattles on about this stuff so much, because it doesn't matter what evidence he hands you guys if you're just going to dismiss it out of hand.
The evidence is incomplete, more so than in other cases, and some use the gaps in our knowledge to make their position seem less ludicrous. But let's not forget how erratic the signal is in South America, and that it has never been detected in many world regions. So while he can keep saying it's not impossible, it seems very unlikely to have anything to do with Hancock's lost civilization. Once again, Mr. Dan the DeDunker tried to extrapolate from a single example without considering all the clues and ignored other cases based on more data that conclusively show we would be able to recognize what Hancock proposes or used to propose, since the backpedaling on how global his lost civilization supposedly was before the alleged cataclysm is probably going to become more and more obvious. When it comes to the limitations or problems geneticists have to face, I have mentioned some, like estimating contributions from groups that are genetically very similar, or the lack of samples, But there is more to say, of course. Dan responded to some of the YouTube comments in the video he removed, and he finally admitted that the evidence wouldn't be missed if it comes from the skeletal record, which is something I told him, but he didn't want to acknowledge. However, very little of what he says there is true, because we actually get a lot from the record, as the samples have been growing exponentially since 2017, reaching more than 10,000 at the end of 2022. I concede that most of them are not from the Ice Age or the Younger Dryas period, but Dan was not giving accurate information, because our modern genomes are not useful to discern if the Upper Paleolithic Magdalenians admixed with Western hunter-gatherers, for example, but thanks to ancient DNA, this has been documented and replicated multiple times by now. We know that Mesolithic Iberians carried high proportions of these ancestries, and that the older Upper Paleolithic signal still survived in some early Neolithic individuals, so the DNA evidence of the lost advanced civilization could be present in individuals who lived thousands of years after the alleged expansion. In my view, what's more problematic is that the vast majority of ancient DNA samples available were collected in Europe and the Asian part of Russia, so testing more individuals from other regions is probably the last hope for those who believe in a lost advanced civilization. There has been a great deal of interest in Europe's past, which partly explains the disparity compared to other regions, but the following should also be considered.
It can seem like there are bazillions of new news headlines and everybody's getting all these genomes and it's working super well. Please understand that in order to get one ancient genome sequenced or characterized, you are going through a bazillion failures in order to get that one precious genome, right? It is most individuals or animals or plants or soil samples do not have ancient DNA preserved at all, or they have such little amounts of it that it is not possible or cost effective to actually try to sequence it. And so it's a very, very difficult field to be in if you're not very patient and if you're not kind of slightly masochistic. It's really hard to work in the lab for 6 months, and at the end of the day, maybe you have like one of the samples you've been working on actually produces DNA.
Yeah, is that a function of the age of some of these samples, or the climate they're in, or just in general?
Yes, all of the above. So
Okay.
age affects preservation, climate affects preservation. So we get the best samples, the best preserved samples are from ancestors who have lived in a cold, dry environment. Generally speaking, there are exceptions, but a cold, dry environment tends to preserve DNA better than a wet, hot environment. And so, and age definitely. And there's other things like what kind of soil bacteria is present and that sort of stuff. So.
Yeah.
And you cannot know in advance what preservation is going to be like. You just can't tell. So some people, and I talked to some archaeologists who think, oh, well, look, the preservation, like this bone looks really good. It's really well preserved, very little mineralization. We should be able to get great DNA from it. I'm like, we don't know. We will
Yeah. I mean, if you think about it, you're talking about wet, hot environments. Some of the best bones I've ever analyzed were from ancient Athens, deep inside wells. And so we're talking a wet, warm environment. The bones are beautiful, though. They almost look like they come from a modern cow or something like that. But the chances of DNA are problematic because of that. It doesn't mean we couldn't, but we wouldn't hold our breath, let's say.
No, and you can still get DNA from bones in that kind of context.
Yes.
It's just, you just can't, the point is you just can't tell until you do it.
I thought that was relevant, and Jennifer Raff explained it very well. So thanks to her and Flint Dibble for sharing good information, and don't forget to watch the video. Next, contamination is definitely an important factor to consider, and to the best of my knowledge, scientists carefully screen for it when working with ancient DNA. Different types of methods are used to do this, which can be classified as follows. Methods based on differences in the DNA sequence, methods based on deviations from the expected ploidy, and methods based on patterns of ancient DNA damage, each being more useful depending on the specific context or type of contamination. However, DNA from other organisms still contaminates most ancient specimens, but the extent obviously matters, and low levels of contamination do not pose a significant problem, which is what we usually observe. Heavily contaminated samples can make reliable inference impossible, and while it is rare for scientists to overlook or ignore this, exceptions do exist, as demonstrated in the recent re-examination of the MZR individual from Southeast Asia by Daniel Tabin and other well-known scientists, a peculiar case that showed unusually high error rates not only attributable to cytosine deamination damage, well documented in degraded samples, but also to previously unobserved patterns that remain unexplained. Normally, even when the source of contamination is not clear, which often happens, there is no problem to identify its signals, but sequences from contaminating DNA that are similar to the reference genome can pass this filtering step sometimes, particularly when studying ancient human material. Human DNA that can potentially lead to false signals of admixture or to underestimation of the divergence to present-day humans is abundant in research environments. But as long as preventive measures are applied and the experts do all the checks, highly contaminated samples passing as genuine shouldn't be a frequent issue. Back to the study covering the samples from Chichen Itza, the first thing some of you probably think is that the European signal detected in this ancient sample is contamination, which is a possibility, but since I couldn't see anything specific on that one, I thought it was a good idea to contact the lead author of the paper. Our conversation was brief and in Spanish, so I'm not going to reproduce it here, but he told me contamination was not the issue, but genome coverage. That's an important factor, because it's obvious that the higher the coverage, the better, and this individual was among the samples not reaching 20,000 SNPs, so the decision was to not remove it from the admixture plot, but wasn't kept for downstream analyses. It is known that low-coverage samples often deviate towards other populations, and sometimes that's all we have to work with, but when it's possible to compare with other samples, the broader context matters to discern whether a signal is real or not, and in this study, the rest of the ancient samples tested show no signs of European admixture, nor do they carry any European haplogroups. So there's important evidence to conclude that the result is not reliable. But even if we assume the admixture is real, our first assumption shouldn't be that the Ice Age Europeans brought it to the Americas. My assumption was that this could be an excess of ancient North Eurasian admixture, an interpretation not incompatible with the low-coverage explanation, which their analysis simply read as European because they were not focusing on the much older signal, and I contacted Daniel Tabin to know his opinion about it. He's a PhD student in David Reich's lab, very knowledgeable on the subject, and he already published research as we've seen. So I thought that a second opinion coming from someone not directly involved in the paper would be also interesting. The specific question was whether a low-coverage sample could show an excess of something, in this case ancient North Eurasian ancestry interpreted as European, that a high-coverage one would not, and his answer was that this can happen using the admixture tool, but not with QPADM, another tool that also calculates admixture, but provides a p-value and doesn't falsely reject models due to low coverage. This means that the tools used also matter, and the test we're discussing was done using admixture. So my assumption was not impossible, but probably not the reason why the sample shows some European admixture. It's also important to note that Daniel Tabin did not play with the data, and that he took what the lead author of the paper said very seriously, but he thinks contamination and low coverage might be causing this, since most if not all ancient samples are contaminated to some extent, and the low coverage ones are basically more affected by it. So this is certainly more precise than what the lead author of the paper told me, who I'm pretty sure is well aware of this, but having found little or no contamination, he likely thought it wasn't necessary to go into more detail regarding that sample. It's easy to understand why low percentages should be taken with a grain of salt in cases like this, and some of you may recall the study I mentioned earlier, where some of the samples tested were very low coverage, particularly the one having very high hunter-gatherer ancestry, which not only shows a higher margin of error, the authors also state that its position in the PCA should be interpreted with caution. That doesn't mean there is nothing to learn here, It just means the results are less precise. And note that unlike the sample of the other study, here both ancestral components are very high, and we have found other early Neolithic samples with very high hunter-gatherer admixture, so it's reasonable in this case to take the results seriously. Finally, new tools and methods are also being developed to get the most out of the ancient DNA samples, like the one described in this paper that analyzes long DNA segments shared between individuals and concludes the Yamnaya culture was the source from which corded-ware groups emerged, or another one published in 2025, which has proven helpful for tracking migrations of genetically similar groups, a task that, as mentioned, has been an important limitation for quite some time. This tool, by the way, further confirmed that there was a punctual admixture event between Neanderthals and anatomically modern humans around 60,000 years ago, settling a long-standing debate.
If we found them, we could test them, we could show that, oh, this group, they were here from over there way before than we expected. How did this happen? Or even like with admixture of like, we could find potentially ancient admixture from cattle into bison or something. If this global civilization was like helping people, they'd be bringing stuff with it. The DNA does not support it at all. And it's just, I don't know. It's just...
It would take years to find multiple samples scattered around the world. And while we've been collecting ancient DNA for some time, more data from certain regions would be valuable before claiming it's impossible that a lost advanced civilization existed. Some silly claims can certainly be refuted, though, and overall the results align with archaeology and other disciplines involved in the study of the past, none of which are finding any evidence either. So it's not looking good for the proponents of the lost civilization, but if we were ever to find Solutreans in the Americas, Australia, Madagascar, and so on, the implications would be undeniable. I hope it's clear that sooner or later, the DNA evidence of the alleged lost advanced civilization that mapped the world during the Ice Age would be found if it really existed, but we'll probably never see that happen. Very often, the work published by archaeologists and historians is crucial for interpreting DNA results, and the more data we have, obviously the better, but as more samples are collected and the methods improve, even the DNA alone is allowing us to draw a lot of solid conclusions. Individuals like Dan or Hancock fail to engage with the current state of knowledge, and sometimes deliberately misrepresent it to promote entertaining narratives, even going so far as to accuse scientists of lying. But aside from creating drama that doesn't hold up to scrutiny, their claims can only persuade the credulous who know nothing about the evidence. I'm not as knowledgeable as the scientists who investigate modern and ancient DNA, but I have a genuine interest in what they do and their findings, and unlike others, I've done my homework to put this together. A lot of time and effort went into this, but it's still nothing compared to what scientists do to publish their studies. Most of them don't have the time to address popular misconceptions in this format, even though some definitely do on social media, so pretty sure they can explain all these things better than me, but I did my best. There is no doubt that the tools and methods developed over the last decades have allowed us to learn so much about our past, and there is surely a lot more to come. Special thanks to the scientists who helped answer my questions, and to those who regularly share their knowledge on this fascinating topic. It really means a lot and makes a difference. Please drop a like, leave a comment, and consider subscribing if you haven't yet. All the best to you all. See you next time.